All right, so I'm going to do a quick lecture about the Western blot procedure, which we're going to be doing. So in class, I kind of mentioned that the whole point of transferring the proteins onto a membrane is that it sort of allows you to do things more easily because the membrane itself, which now contains kind of like a photocopy of the proteins that were run on a gel, the membrane offers you sort of like a stable, robust kind of element that allows you to just sort of work with in a lot more easier fashion. Definitely more so than working with the gel, which is kind of like thin and it can tear and things can diffuse through it and so on. So anyway, in terms of the actual Western blot, I'm going to go over the classical Western blot procedure, but just sort of pin it for now that the procedure that we'll be doing in class is actually called a quick method that was actually developed by Millipore. And I'll kind of get into that in a little bit more detail actually during class. But anyway, to kind of set up how things work in the classical procedure, what I'm going to do is I'm going to attempt to kind of doodle the sorts of things that are going on. So firstly, we did the transfer. So here we have a membrane that I've drawn. And basically the transfer allows us to actually attach proteins to the membrane. And if you recall, I mentioned that the primary kind of thing that's getting the things to bind to the membrane is actually hydrophobicity, right? Especially at the beginning, where we basically force the proteins using an electric field onto that membrane. And you can imagine that the proteins that are now on the membrane are going to be a mixture. There's going to be proteins that are actually things that you are interested in, right? Like this is the actual thing that you want to see. And in the case of a Western blot procedure, this is the thing that you actually have an antibody that can specifically target this protein of interest. Because of course, you're also going to have a whole bunch of other proteins that are in this scenario that also attach to the membrane. Okay, so this is sort of the setup that we have at the end of class. And when we begin the Western blot procedure, in the classical context, we have to make sure that the membrane stays wet, or if it doesn't stay wet, that you do go to the effort of wetting it. And because we're using PVDF or a nylon-based membrane, that will usually involve a step involving methanol of some sort, okay? So let's assume that our membrane is still wet. And just a side note, we're drying our method, but that's got something to do with the quick method, actually, that we're actually doing in class. So we have a wet membrane. And usually the very first step that needs to happen is something called a block step. And so basically a block step is a scenario whereby you are going to have the solution that usually has this kind of recipe. It's usually 5% BSA in TTBS, okay? So a couple of things I want to point out. TTBS you're going to see over and over again, and TTBS basically stands for tween 20 tris buffered saline. Okay, and so basically this is your basic wash buffer. Okay, it has a little bit of tween 20, which is a weak detergent. Okay, and so that is really good at basically dislodging anything that might happen to be non-specific. And it has tris, so you pH it, and it's basically at a physiological pH. And there's also a bit of salt just to make sure kind of like the ionic environment is friendly to proteins in general. Okay, the key thing here though is that there's this BSA, and BSA is short for bovine serum albumin. And basically the idea here is that this is acting as a generic protein. Okay. And the reason why you want this is because you need a generic protein of some sort to essentially cover the rest of the membrane. This is why it's called a block step. Okay. And the reason why you want to do that, I think is as you can intuit, is that if you don't do that, right, you have all of this exposed membrane, which if you remember is like super hydrophobic and just ready to bind to anything that's hydrophobic as well. And so if you don't do a block step and you come in with an antibody or what have you, that antibody is going to be perfectly happy binding to the exposed membrane. And so that generally just results in a lot of non-specific binding, right? So this is what the block is fulfilling. It's covering the exposed parts of the membrane so that you don't get that non-specific binding, okay? And bovine serum albumin just happens to be a very common generic protein that people use because you can buy it in large quantities at a research grade level, because it's often used for things like tissue culture. But that being said, you can use any protein, right? Hypothetically, right? Another very common one is for folks to use skim milk powder. It's sort of like a cheap hack for generic protein. I mean, technically, I could like cut my thumb off and grind it and throw the protein of my thumb onto, as my block solution, because that's just acting as generic protein that can cover the rest of the membrane. Now, the thing to keep in mind, however, is that you sometimes need to think a little bit about what this generic protein is, right? So for instance, I jokingly mentioned just cutting my thumb off and grinding all the protein there. But does it make sense that if my antibody coming in at the subsequent steps is an antibody against, say, human protein, then you kind of run the risk of cross reaction, right? So even though you've used my thumb protein as a generic, quote unquote, blocked protein, if my antibody happens to be specific for something in that block solution, then it will also bind, right? And just FYI, skim milk powder, it does have certain proteins that, depending on what you're working with, there could be that cross reaction. So that's why sometimes you do see people stick to BSA, because it's just the one protein, it's highly pure, and it's research grade and things like that. Okay. So anyway, that's the first step. This is usually about a 2-hour incubation. Okay, but you can also let this go overnight. Okay, so that's the first step. Oops. Now, after that, okay, there's usually a wash step. Okay, and so wash steps for this particular part, because really the only thing you're washing is this block, this generic protein. This wash step is kind of like pretty minimal. It's usually one wash with this TTBS. And normally you would maybe do it for about 5 minutes. Okay. Now, after that initial wash step, then you can kind of get on to the business of actually the antibodies. Okay. So the first antibody step, so this is often called the primary antibody step. Okay. And this is kind of special from the following context. So this is basically where, you're going to have an antibody that is specific for your protein of interest. And of course, because it's specific, the idea is that it's not going to bind anywhere else. Okay, so a couple of sort of logistical things about this primary antibody step. So firstly, you often see this as some sort of dilution, and it also happens to be in that same block solution recipe. So you have a 1 over something fold dilution of the antibody, right, in your 5% BSA TTBS or 5% skim milk powder or what have you, block type solution. So this number here, the dilution is kind of giving you a sense of how good the antibody is. So for primary antibodies, it's not uncommon for the dilution to be somewhere in 1,000 or 2,000. So in other words, you're diluting it 1,000-fold or 2,000-fold or thereabouts. If it's a really kind of like dilute antibody or kind of a crappy antibody, then sometimes you see lower numbers like 1/100 or 1/200. And if it's a really like kick-ass antibody, you might even see much higher numbers like 1/10,000 or something like that. Okay. But quite often they tend to be sort of 1/1,000, 1/2,000 or thereabouts. Okay. This step also usually goes for about 2 hours. Normally you don't tend to go more than that, right? So unlike the block step where you had you could let it go overnight, for instance, with the primary antibody step, you generally stick to that 2-hour time frame. Okay. Now, the thing I do want to point out and talk a little bit more about is like, where do you get this primary antibody from? So again, this is literally the antibody that is specific for whatever it is you're interested in. And, if you're working in a lab and that lab happens to have some focuses and, it focuses on certain proteins, then, actually having a number of these specific antibodies is going to be a super useful tool, arguably a necessary tool for a lot of protein work, so that you can actually monitor whatever this protein of interest happens to be. So I do want to just spend a little bit of time and kind of talk about where you can get this primary antibody, how to get a primary antibody. Okay. So one option is that you just buy it, right? And so, for instance, if you're working in a field that's quite trendy, cancer research, for instance, a lot of the proteins involved in cancer signaling, there are commercially available antibodies that you can quite easily buy, right? If you're working on something that's a little bit more niche, this might be a harder option. And generally, if that's the case, you will have to resort to making it or going through a service that will make the antibody for you. But I kind of thought I would sort of highlight, how you would make an antibody, okay? And there's generally two ways that are most commonly used. One is to make a type of antibody, which is called a polyclonal antibody. And the other one is to make a type of antibody, which is called a monoclonal antibody. And let me just spend a moment to kind of go through these two different options. Okay. So let's start with the polyclonal antibody. And firstly, what I mean by polyclonal is that this antibody reagent is actually a population of multiple different antibodies, hence it's polyclonal, that just happen to recognize your protein of interest at different sites of that particular protein. Okay, and generally how you make this is kind of as follows, right? So first you start with your antigen, okay? And this is just a fancy word for the thing that you want the antibody to be against. So for example, the protein, normally you would have this in a purified form. And essentially making a polyclonal antibody involves taking this antigen and essentially injecting it into bunnies. This is my bad picture of a rabbit, okay? And so the idea here is that you're trying to get the bunny to mount an immune response to whatever foreign substance, in this case, the antigen protein of interest that you've injected into the rabbit. And not only that, but you inject in a sort of a strategic fashion. There's normally booster shots so that you can kind of guide that rabbit to not just mount an immune response, but to actually have the secondary response where you have an elevated production of antibodies, okay? And then after you sort of done this, then essentially you sacrifice the poor rabbit and you take its blood, right? And so from the blood, what you'll actually do is that you'll separate the serum out of it. So that's sort of the blood component minus the actual blood cells, like red blood cells, for example. And, it's sort of this yellowy fluid, but this is where the antibody generally resides. And you literally just use this, okay? Because the idea, again, is that this rabbit has made, a B cell response with all the antibodies. It's secreting the antibodies into the blood, as part of that, active immune response. And essentially, you're just pulling the serum out. And you use this for your antibody solution. Sometimes you can use it neat. So in other words, you can just use it directly because, you know, the rabbit has done such a great job making this immune response. Sometimes it might be a bit more enriched. So, you know, you can do things to concentrate the antibody within that solution. You can do things to pull antibodies out of that solution. But ultimately what you have here is you do have this sort of polyclonal mix, which is just saying that you have a bunch of different antibodies that may recognize all sorts of different sites within that protein of interest, okay? Now, the thing about this method is kind of as follows, pros and cons. So one big pro is that it's relatively easy to do, okay? It's not hard to have a scenario where you get a rabbit to sort of make this antibody solution for you. The second huge advantage of a polyclonal setup is that generally you will probably get something that is useful. And the reason why I say that is because you're kind of hedging your bets with the sample, because it's not just one specific antibody, it's multiple specific antibodies. And in that population of, you know, polyclonal antibodies, there's probably different antibodies that can do different things, right? Some might be good for immunoprecipitation, some might be good for Western blot, and so on and so on and so on. So you kind of hedge your bets because you kind of have a population of antibodies that are specific for your protein of interest. And that's something that usually means that there's probably something that you can get out of that particular mix, okay? The disadvantages of a polyclonal antibody setup is really the fact that it's a one-time use. It's a one-time scenario, right? So if you get a great batch from that particular rabbit, if once you run out of that reagent, you know, you can sort of redo it, but there's no guarantee. And in fact, it's very likely, if not always, the case that that new antibody batch, that new polyclonal batch in this other scenario is not going to be the same, right? And so you sort of lose on sort of the reproducibility side of things, and that can be problematic. Okay. The other thing is that because it's polyclonal, right? Sometimes it's actually useful to know that there's just one antibody and that it just binds one place. The fact that it's polyclonal and you don't necessarily know what it's polyclonal against, like what parts of the protein it's binding specifically, that vagueness can sometimes make interpreting data a little bit harder. Okay, but that being said, it's a very common tool and reagent, primarily because it's just easy to make. Actually, there used to be a guy on campus, his name was Willem, and he used to be like the rabbit whisperer. And sometimes, you know, if you happen to know him, you would get him to help you make the polyclonal antibodies, because he had this sort of rabbit whisperer ability to be able to find the lymph nodes without, you know, like actually hurting the rabbit. So when he would inject the antigen to sort of mount this immune response, he would just sort of intuitively know where the lymph nodes were in this poor rabbit. So just so you can get like a really strong elevated response. Anyway, that's polyclonal. The other one that I mentioned is monoclonal antibodies. Okay. And so, and this is actually, if you were to buy an antibody, it's probably a monoclonal antibody. But let me just kind of tell you how to make a monoclonal. So basically, again, you have your antigen, and this time actually you tend to use mice. Okay, so you inject the antigen into the mice. You basically get the mouse to mount an immune response. Just like with the rabbit, you know, you have a booster shot and all of those things. And then at the end of that immune response, you actually isolate the spleen. Okay. And so the spleen is this sort of oblong, black looking thing. And the reason why you want the spleen from the mouse, and obviously you sacrifice the mouse, is that the spleen is actually a major site of B cells. Okay. Because again, B cells are what make antibodies, right? And so the idea here is that because you've made this mouse go through this immune response, this spleen should ideally have a whole ton of B cells that have been activated to produce antibodies against whatever your specific antigen is, okay? Now, these B cells, what you then do is that you combine B cells with something known as a B cells with something known as a myeloma cell. Okay, and actually you use reagents like PEG, or just polyethylene glycol, which can kind of facilitate cells fusing together. Okay, and so the reason why you want to do this is because you want to create something known as a hybridoma. Okay, and so a hybridoma is literally a dual identity cell, which was created by taking a B cell fused with this myeloma cell. And the reason why you want to do this is because each of those two cells, the B cell and the myeloma, has an element of functionality that you want, okay? So B cells you want because it's making an antibody, right? That's the whole reason why we're here. Problem with B cells is that they are a pain in the ass to culture. So there's no really good way or easy way to actually, let's say you have a B cell that's making the antibody that you want. There's not actually a really good way to maintain that B cell, right? It will eventually die off, right? A myeloma cell, on the other hand, which is basically just a cancer cell, that's all about ease of growing and, cell division and all of these things. So the idea of a hybridoma is that you combine those two functions. You combine the function of a cell that's capable of making the antibody, which you care about, but with a cell that just makes culturing way easy, okay? So you can imagine that if you have this spleen with millions of B cells and you add millions of myeloma cells and you sort of do this experiment to facilitate fusion of cells, that you're going to get a mix of, B cells, two B cells that are fused together, two myeloma cells that are fused together, and sort of a mix of scenarios where randomly B cells that fuse with myeloma cells. So this hybridoma sample is actually kind of a mix, right? So it's a mix of all sorts of different fusion events. And so this then takes you to a part of making monoclonal antibodies that's actually quite labor intensive. So you sort of have this mix of fused cells. And again, the idea is that in that mix somewhere are hybridomas, and not just hybridomas, but hybridomas that happen to be making antibodies that you care about. So what you actually do with this sort of mix, kind of this ugly mix of potentially fused cells, is that you actually plate them out on 96 well plates, right? Where each well has a little bit of the antigen, okay? And so the idea here is that you take this mix of fused cells and you dilute it out so that when you dispense it into the 96 well plate, you sort of done the math so that the plate is either going to be, like the solution is so dilute that you can guarantee that only one fused cell has made it, or no fused cells has made it. So it's just you're just basically diluting it so that in every well it's either nothing or just one cell, right? And basically, because there's an antigen on the bottom of that well, in that 96 well plate, if there is a hybridoma with an antibody specific for the antigen, it will activate it and it will get it to proliferate. So basically, when you make a monoclonal antibody, there's this very high maintenance, a lot of work, monitoring step where you have these 96 well plates, and you might actually have 20 to 30 to 40 of them, right? And you sort of spread out your potentially hybridoma mixed in it, and you're essentially looking for growth, right? Because if there's growth, that infers activation of whatever was in there to grow and multiply. And because it can only grow and multiply because it's interacted with the antigen, then the idea there is that, oh, I've got a hybridoma, and not just any hybridoma, but a hybridoma specific for my particular protein of interest or antigen, okay? So it's a lot of work, but the thing is, and this is the huge advantage of monoclonal antibodies, is that if you have that hybridoma, you have an unlimited like cell factory making this antibody for you, right? So you know, it's a lot of work to make. It often doesn't produce anything, but if you do get something, you literally have something that you know, that will give you an endless supply of a specific antibody. Okay, so that can be very, very good. And you know, because it's monoclonal, there's a lot of advantages to that. So one of them is that because it's monoclonal, like literally one antibody specific for one site on that antigen, you'll probably go to the effort of trying to figure out exactly what epitope it's binding to. And if you do that, then you can actually begin to do some more finessed experiments with that type of antibody, you know, like inhibition experiments and things like that. So it can be very useful to know that it's not sort of like a random population of things, but just the one antibody specific for the one place. Okay, so yeah, so that's you know, I guess the pro is that it's like woohoo if you get one, because you know it's like an endless supply. But the con, of course, is that it's a lot of work and often without success when you're actually trying to make it. Okay, so there are the two types of antibodies. Sort of a long side tangent talking about this step, but this was the primary antibody step, and again, this takes about 2 hours and thereabouts. Now, after that, there's another wash step. Okay, and this wash step is usually, I find, what people hate the most, because it's taking your TTBS solution, right? And you're going to incubate for 5 minutes, but you're going to do this 6 times. Okay. And so it's just sort of a pain because, you know, all 6 steps total a fair chunk of time, like a significant amount of time. But at the same time, because you have to change things every 5 minutes, it's not enough time so that you can, you know, do other things. But that being said, you do want to do the wash step, because what you're trying to do here is make sure that you don't have any non-specific binding of your specific antibody to other places. Okay. Now, after this wash step, there is the secondary step. Okay. So let's talk about the secondary antibody step. Okay. And essentially what this is that this is an antibody that is specific for the constant region of the primary antibody, just as I've drawn here. And also usually there's some sort of means of detection involved. Okay. So again, this is often labeled as a dilution, you know, 1 over something. And because secondary antibodies are almost always commercially bought, they tend to be very good. So that X number tends to be very high, like 1 in 10,000 or something like that. But it too is still in its, you know, the blocking type recipe, you know, 5% BSA, 2TBS. Okay, and this is usually about a 1 hour incubation. Okay. Now, let me say a few things about this secondary antibody, like what it is exactly. As I mentioned already, it's something that binds the primary, but it also is something that has a way of detecting. And because of that, what you tend to see, you know, how they're labeled, they tend to be like an organism anti-organism IG type. So what I mean by that is that you might see a label that says it's goat anti-rabbit IgG. And what that means is that you've picked this one because your primary antibody was made in a rabbit. It happens to be an IgG type. And just so you know, the secondary itself was made in a goat, right? So it tends to have this sort of nomenclature, okay? And now alternatively, You might also hear of things like protein A or protein G. These are actually secondary systems, but they're not technically antibodies. They're bacterial proteins that have specificity for the constant regions of antibodies. And where they can be sort of useful is that unlike a conventional secondary antibody, which is very specific for the type of isotype and the organism it came from, these bacterial proteins, which are also recognizing the constant region of antibodies, tend to have a palette. Like there's a number of different options. So for instance, you might be in a lab and you have 5 different primary antibodies, but they're made from all sorts of different sources. So you can actually buy 5 different secondary antibodies specific for each primary antibody, but you might actually find that the protein A palette can recognize all of them. So you can just buy the one reagent that can be used for all of them. So it's that sort of thing. So that's why sometimes you see protein A or protein G. The other thing that often comes up is, what is this means of detection? So this is usually an enzyme of some sort, right? Alkaline phosphatase is quite common. These days, horseradish peroxidase is probably more common. And the idea here is that if you have this enzyme will later on be able to drive sort of like an enzymatic substrate sort of reaction, okay? And so it kind of depends, means of detection, like in the past, sometimes there were ways to radioactively label the antibody, but anything basically that allows you to see that the secondary antibody is there, okay? One thing I do want to point out is that sometimes a question is asked, why even need the secondary antibody? Why can't we just label the primary antibody? The reason for this is twofold. So firstly, it's not easy to sort of attach an enzyme onto an antibody. So it's high maintenance. So it's much easier to just be able to buy these commercially available secondary systems. The other thing is that with this 2-tier antibody system, you can actually get very good amplification of signal. Okay, so it'd be sort of like, let's say I'm the primary antibody and I am anti-screen, right? And I'm waving a bell, right? So if I'm labeled, then the screen is there because you can hear the bell, right? But if you can imagine that all of you are anti-Dave and you put your hand on me and you all have bells, do you see that by having that 2-tier system, you can greatly amplify the signal? So this is something I do want to mention, is that Western blots, generally speaking, are super sensitive. So they're great for being able to detect very small amounts of proteins. But at the same time, one of the disadvantages of this sort of 2-tier signal amplification is that it's very difficult to get a quantitative read on what you're looking at. So in other words, if the band is twice as dark, that doesn't necessarily mean there's twice as much stuff there. So that's something that you need to keep an eye out for. But in any event, the secondary step usually, as I mentioned, goes for about an hour, right? And after that particular step, then there's another wash. And this is an important wash, right? Because again, you want to make sure that there's no non-specific binding of the secondary antibody. This other wash is again this super annoying 6 times 5%, 6 times TTBS for 5 minutes. Okay. And then you sort of close in with whatever substrate step comes in next, right? So i.e. the substrate that you add to the mix, because there's the enzyme there, it gives you the product. And this is what is detectable. Okay. And again, the idea here is that you're only going to see a product, right, where the substrate was able to be worked upon by the enzyme. The enzyme is only there where the secondary antibody is. That secondary antibody is only there because it's binding to the primary antibody, and that primary antibody is only there because it's binding to the protein of interest, right? And hence you get your signal. So just so you know, probably the most common sort of substrate system right now involves ECL or chemiluminescence, which is often driven by horseradish peroxidase. We're actually going to use a different scenario. It's still HRP, but it's actually driving the production of a crystal dye that's sort of pink in color. And the only reason we're doing that is mainly so it just saves us a trip from crowding a dark room and trying to look at our data that way. Okay, but basically, you know, this is the classic procedure. And again, I just want to highlight that we're not doing it. We're doing something called a quick method, but this classic procedure would tend to take a minimum 6 hours, right? And this is sort of the normal way of doing it. Okay, so I'm going to stop here, we will talk a bit more about the quick method when we're actually doing it in class. Okay, bye.